Welcome everyone. I'm Mike Fave, future nurse practitioner and independent health researcher. In my last video titled The Dynamite Spark Duo That Ignites Heart Disease, I highlighted the interplay of reactive oxygen species and polyunsaturated fats in the development of cardiovascular disease. Today I want to hone in specifically on the mitochondria, one of the key sites of energy production in the body, as well as ROS production, and give you an idea of how ROS are intimately involved in fine-tuning mitochondrial metabolism and how this ties directly into not only your heart health, but your health in general. So with that in mind, let's jump in. So to start, I'm going to be discussing essentially two papers in this video. The first paper I have cited here, it's titled Role of Mitochondrial Reverse Electron Transport in ROS Signaling. And then the other major paper that I have involved in this video is called Cysteine-Mediated Redox Signaling in the Mitochondria. So if you guys want to check those papers out, you'll be able to do so. I have all of the sources listed here. And I can link those papers inside the description. So the first thing that I want to share with you in this video is that mitochondria are the main source of reactive oxygen species generation for most cells. And if you didn't watch my previous video discussing the interplay of reactive oxygen species and omega-6 polyunsaturated fats in cardiovascular disease, I recommend going and checking that out because it gives the underlying basis for why I'm actually creating this video today. So the first quote I want to share, and it's just to lay the groundwork as we start to go through the rest of these processes, is this quote here. And the quote states, mitochondrial reactive oxygen species produced by the respiratory chain during oxidative phosphorylation are the main source of free radicals in most cell types. So across the body in general, as we discussed in my previous video, one of the key sources of reactive oxygen species production is actually the mitochondria. And if we look here in this graphic, we can see that at all of these areas where you see the stars is areas where reactive oxygen species can be generated in the mitochondria. And it kind of makes sense. This is essentially a controlled combustion to a large extent happening within the cell. And so there's a lot of opportunity for there to be an electron leak that can interact with different components inside the cell that can lead to the formation of these reactive oxygen species. So this is one of the key sites that this occurs, and this is why I'm actually discussing this. But also in a larger context, this highlights how important energy metabolism actually is in a variety of disease processes, because energy metabolism is directly fine-tuned and adjusted by ROS signaling. And we're going to get through that throughout the course of this video today. But essentially, the ROS production, the site of production, as well as the amount of production, is going to give us an indication of what's going on metabolically. So the cell is using the signal of ROS to say, hey, are things going well? Are things going poor? What's kind of going on? So that's one of the ways that it's fine-tuning its metabolic rate. And so in states of stress, cardiovascular disease, heart disease, you're essentially starting to see the overproduction of ROS, and it's a signal to the cell that there's problems metabolically. So that's the frame that I want to discuss this video in today. So let's continue on. And so just as I was mentioning, not only ROS amount, but the site of production is important. So I'll read this quote and then we'll get into what this actually means. So the current consensus is that the amount of ROS determines their physiologic effects, with low levels of ROS having beneficial effects through stimulation of mitohormesis and high levels causing oxidative damage and contributing to aging. In the last 10 years, the development of new technology and methods has allowed the measurement of mitochondrial ROS levels in more physiologic conditions. Evidence collected using these techniques indicate that the site at which reactive oxygen species ROS are generated is as important as the amount of ROS produced. So the overall picture here, that it's not just about amount of ROS that's being generated. It's not just this pure gauge of, oh, if we have a little bit, it's okay. And then if we have a lot, it's a problem. What they're starting to actually see is, no, it's actually, where is this ROS being generated inside the mitochondria that is dictating what the outcome is from that. So that where the ROS is being produced is sending a signal to the cell through a variety of mechanisms. We'll discuss some of those mechanisms that says, hey, metabolism is here, metabolism is there, and then the cell can start to fine tune its different processes based on the signal that it's seeing. And on a point to the mitohormesis argument, or the hormetic argument in general, I think that this next layer of understanding of ROS signaling actually kind of contradicts the idea of mitohormesis. The general idea of hormesis is that a small amount of some toxic substance induces a benefit through like a protective or defensive response from the organism, whereas a larger amount of this protective substance can kind of or this damaging substance, excuse me, can override the cell's ability to deal with it and actually causes dysfunction. And so that's been a theory or a mechanism that's been used to describe a variety of different processes. But essentially, I don't think that this is a hormetic effect. I don't think that just a small amount of ROS is inducing this protective effect and then a large amount is just crippling the cell. Like, that does happen. What I think is more likely is that the amount of ROS generated at particular points inside the respiratory chain, so you can kind of see that here, inside the mitochondria, lets the cell know how things are functioning. And so it's more that the ROS is a way for the cell to gauge what's going on and less so just this basic hormetic idea. So it's more a general idea or a way to create an analogy that makes sense here is that if you think of the mitochondria as an engine, the amount of smoke that's emitted by the engine, it indicates how well the engine's functioning. Or the temperature of the engine, or the number of RPMs, the repetitions per minute in the engine, give you an information as to what's going on with the engine, how well it's functioning. And so I think ROS can be considered to some degree a factor like that, where it's a way for the cell to get a sense, and also the mitochondria itself to have this process, this feedback loop where it can get a sense of how much ATP is being generated, how much ROS is being generated, what's going on with the proton motive force across the membrane, so how much protons are on one side versus the other side. And so it's looking at all these factors, and then it can adjust and fine-tune its metabolic function and rate and bring other things on board, say antioxidant enzymes if necessary, to deal with whatever is going on metabolically. And like a regular engine does the same type of process overall, where basically you're the pilot of the engine, or the engineer who's working with the engine, is looking at different indicators for the engine's function and then can tweak different components to optimize the engine's function. Because when you're producing energy in any extent, you're going to get some heat, you're going to get some smoke in the engine idea. It just what's more important is how much of those things are going on, are indicating the function and ability of that particular engine. I think ROS is that signaling inside the mitochondria, and it's not necessarily this hormetic piece. Again, it's more that this is signaling and indicating how is the mitochondria actually functioning. And I think that's a huge perceptual shift for anybody who's been following any of the hormetic research, because it kind of turns some of the hormetic ideas on their head. Instead of like, oh, we just need to induce this defensive mechanism, and becomes more of something where the cell is actually looking at these different components and trying to adjust its function based on the information that's receiving on a regular basis. So I brought this picture back again just so you guys could see there's multiple paces inside the mitochondria where you can actually get ROS production, and where you produce this ROS, we'll get to see that throughout this video, whether this is complex 1, so whether it's this side of complex 1 or this side of complex 1, or if it's at complex 3, or if it's related to something in complex 2, or if it's happening in some of the different enzymes functioning in the citric acid cycle. Those are all different indicators to the cell that are saying, hey, there's something going on here. And it's a location effect. So is it happening in complex 1 versus complex 2? And then it's an amount, like how much is being produced. There may be an amount that is okay for complex 1 to function. That's normally understood to be like, that's a normal function or normal ROS amount given a function for complex 1. But then at the same time, at a certain threshold, it starts to signal perhaps the system is breaking down, or efficiency is adjusted, or something along those lines. So I just use the same graphic to kind of show that idea. Now, the next piece, so as I was discussing, is the ROS is a signaling molecule. Now, what exactly does this mean? Like, how does it go about signaling? So there's multiple ways that ROS goes about signaling, but this is just giving you guys an example of one of the pathways in which it can induce signaling. What is the actual mechanism that can underlie this? And again, this isn't the be all to end all signaling mechanism, but it is one signaling mechanism to think about. So in the first piece here, the quote says, however, an increasing amount of evidence has shown that mitochondrial ROS play a more complex role than previously anticipated, actively participating in cellular signaling. For example, hydrogen peroxide, a stable reactive oxygen species, is able to diffuse far away from the mitochondrion and alter the activity of proteins by regulating the oxidative state of one or more cysteine residues. This type of redox regulation has been shown to be instrumental in controlling many physiologic processes. So these researchers actually reference the back in Waropana article, and then so I went to the article and I pulled out this graphic describing this process. So as what we can see here on the bottom is we have hydrogen peroxide, H2O2, and you can see this is a gradation in the amount that's actually being produced. So in lower levels of hydrogen peroxide production, you have the periredoxin protein, and then the periredoxin protein has different cysteine groups on it. And the cysteine groups, the sulfur component of the cysteine groups can react with the hydrogen peroxide. So basically the only thing you have to consider is there's a protein here, and there's a particular part of this protein that directly interacts with hydrogen peroxide. So the protein is kind of like a litmus strip of how much hydrogen peroxide is being produced inside the cell. And as this protein, as the cysteine group in this protein continually gets adjusted by larger and larger amounts of hydrogen peroxide, eventually the protein becomes non-functional, but along the way, it gives an indication to the cell like, hey, that we have X amount of hydrogen peroxide going on. And it's based on the change in structure and function of this protein via the cysteine's reaction with hydrogen peroxide. So I'll read you guys the general idea here so you can get this. I'll read you guys the information here so you get the specifics, and we'll walk through it. But that's the general idea. Essentially, the proteins are functioning as litmus strips inside the cell based on their changes to their structure from reactive oxygen species like hydrogen peroxide to get a gauge on what's actually going on inside the mitochondria. So what they say here is hydrogen peroxide induced inactivation of peroxireductins to generate an oxidative burst. So under standard hydrogen peroxide levels, the peroxidatic cysteine of peroxireductins, so this is a protein, reacts to form a sulfenic acid. So that's the sulfur and hydroxide group. So you can see that here. So this is the peroxireductin protein. This is where it has the cysteine group, the peroxidatic cysteine group, and then when it interacts with hydrogen peroxide, you get the formation of this sulfenic acid here. And so this protein structure has changed a bit in response to the hydrogen peroxide. Now, what they talk about here, they say that the sulfenic acid, which is converted to an intermolecular disulfide. So these things, the peroxireductin proteins can form disulfide bonds through this group, and then they kind of form this dimer. So they're interacting together. It's not as important as the general idea, but that does happen. Now, what they say is disulfide formation is relatively slow due to the requirement of conformational rearrangement. Disulfide reduction is promoted by the NADPH-dependent thyroredoxin reductase, so TRX to TRXR system. And upon increased levels of hydrogen peroxide, the peroxidase activity of peroxireductin is attenuated by hyperoxidation of the peroxidatic cysteine to form a sulfenic acid. So what happens is in the first step with more moderate amounts of hydrogen peroxide, you get this sulfenic acid group. And now the cell has an ability to convert this sulfenic acid group back into the normal peroxidatic cysteine group on the peroxireductin protein. So it changes this back into this. Now, if the hydrogen peroxide levels continue to increase, then you actually start to form sulfinic instead of sulfenic. So that's where you see the SO2H here. And what you can see is that now this is another indicator. Okay, there's more peroxidative stress going on. You have larger amounts of hydrogen peroxide being produced, and it's changing these proteins. So at this point, the cell, and what we're going to start to see here, still has the ability to convert this sulfinic acid back into the sulfenic acid and control, some of the oxidative stress. Now, and so I guess we'll just read that first. So formation of this hyperoxidized species is possible due to the extended half-life of the sulfenic acid species. This inhibition of peroxidase activity allows for a buildup of ROS for signaling purposes. The sulfinic acid can be re-reduced by ATP-dependent sulfur reduction. At extreme levels of hydrogen peroxide, the peroxidatic cysteine is irreversibly overoxidized to sulfonic acid. And so essentially, at a certain point, the cell is unable to actually deal with the oxidative stress response from the hydrogen peroxide. And then you get the formation of the sulfonic acid and the overoxidized cysteine group on the peroxide reduction. It completely changes the protein structure, and its function is also adjusted due to that change in structure. So again, the way you can think about this is this is a litmus strip. And so if anyone's ever had a pool or a hot tub, or they ever check the pH of something, you essentially dip the strip in water, and then you get different colors in terms of the acidity or the chlorine levels or something along those lines for that water or whatever you're testing. And so the way the body does this, its litmus strip is these proteins. And so when hydrogen peroxide is increasing to very high levels, it changes the residues, the cysteine residues or cysteine components of these proteins. And then what winds up happening is the function of those proteins change. And this sends a signal overall to the cell to say, hey, there's something going on here. Now, what we're going to start to see in the next picture is what does this actually look like inside the mitochondria. So here we have a graphic. This is complex 1 inside the mitochondria, and it's discussing a couple components. So the changes to the peroxide reducin proteins and the changes to the cysteine group in response to hydrogen peroxide is one mechanism. They're going to talk about another mechanism here, which involves nitric oxide. And so let's go through this so we can see what does this actually look like in terms of changing function inside some cellular function in the mitochondria. So we have here, they say protective effects of cysteine nitrisation and glutathione on complex 1. So complex 1 accepts electrons from NADH. So we know that it takes NADH, converts it to NAD+, and this is where the NADH and NAD+ ratio become important, and also NADH to FADH2 ratio, which we'll get to. And then it transfers these electrons along a wire from the FAD, so it's a flavin group, to 8 iron sulfur clusters, and ultimately to a quinone group. Under standard conditions, low level of ROS, or I guess before we get to that, so NADH drops off some electrons to this FADH group, and then the FADH transfers these electrons inside complex 1 around this, essentially think of it as a wire, so these iron sulfur clusters to this quinone group, and then this quinone group should, if I remember correctly, should transfer the electrons to coenzyme Q, which we'll see in just a second. So the normal process, you have electrons dropped off, and you can think of this system, it's like largely an electrically driven system. So electrons are dropped off through to the FADH group. They travel along this wire of iron sulfur clusters, which reach a quinone group, which drops off the electrons to coenzyme Q. So what they then say is under standard conditions, low levels of ROS are generated at either the quinol, so we see that here, or the FAD sites. But under conditions where levels of NADH are high and electron flux is low, significant and damaging levels of ROS can be produced. Glutathionylation of 2 cysteine residues near the flavin binding sites results in reduced enzymatic activity and a decrease in ROS generation, possibly by excluding NADH binding and electron transfer. So I guess here is the normal function. This one has the nitrosylation or nitrosation. So NADH drops the electrons off at this FADH group. They travel along this iron sulfur clusters or the wire to this quinol group, and then they drop the electrons off to coenzyme Q. As they do that, they move protons across the inner membrane, the inner mitochondrial membrane. We see that here. And then now say there's a problem where you essentially have high levels of NADH and then electron flow is low. So the electrons aren't flowing appropriately. So where would something like this happen? Well, if you have an excess of fatty acid oxidation or large amounts of succinate going in and producing higher amounts of FADH2, then coenzyme Q here would be essentially leading to a backup here at complex 1 because it would be over reduced or it has carrying too many electrons from complex 2. And so what can happen then is that you have these high levels of NADH and the electrons aren't able to flow. There's nowhere for these electrons to go. And so then you start to get production of ROS. Now, what happens is the ROS can trigger a glutathione response. So glutathione is an antioxidant. And so the glutathione would interact with you can see here, these would be the cysteine groups. So it interacts with the cysteine groups. And essentially, when the glutathione interacts with the cysteine groups, in order to protect the cell from the ROS being generated, because the NADH can't drop off electrons, it inhibits the FAD group from binding to the NADH's electrons. So the NADH can't drop off the electrons here anymore. So now you're seeing a process where you have an oxidative stress from these electrons lead to changes inside the cysteine groups. Then you have the cysteine groups have a glutathionylation. So glutathione comes in and binds with the cysteine groups, protects them from the oxidative stress. And this change in the protein structure from the addition of the glutathione inhibits the electrons from being able to bind into the FADH here. And what winds up happening is now NADH, again, can't drop off these electrons. So it protects complex 1, but it shuts down electron flow through complex 1. And this is adjusting via the oxidative stress. So you can see how the oxidative stress is creating a signal to the complex 1 proteins that, hey, things aren't working well now. And so then you have this response where the glutathione comes in response to some of the oxidative stress, binds with the different cysteine groups, and then when it binds to those cysteine groups, the protein stops transferring the electrons effectively. So that's one scenario where we're seeing it function as a signal. And in the alternate scenario, they say alternatively, nitrization of a different cysteine prevents complex 1 reactivation during ischemia reperfusion, preventing a burst of ROS production from a rapid reactivation of the complex. So when mitochondria or the metabolism, the metabolic structures inside the cell are under significant stress, there is a change in nitric oxide production or levels or concentrations. And the change in nitric oxide concentrations can affect what's going on metabolically, can affect the different components of the respiratory chain. For example, it can affect the cytochrome C's function to some extent. There's more specific mechanisms on what's going on there, and I don't want to go into them for this video, but you start to get this increase in nitric oxide. And so when there's higher amounts of nitric oxide can easily form reactive nitrogen species, which have similar effects as reactive oxygen species. And what happens is the nitric oxide at certain concentrations will come in. It serves as a signal, hey, there's something going on here. And it will interact with a cysteine group on complex 1 inside the mitochondrial membrane. And what winds up happening is it stops complex 1 from reactivating for example, in these stressful situations like ischemia reperfusion. And the reason this is actually helpful, because if complex 1 was reactivating, you just had all this NADH lying around, what could happen is you can just get large amounts of reactive oxygen species production because things aren't going to flow through the electron transport chain effectively, and you have the large amounts of NADH ready to drop off these electrons. So it's actually a protective mechanism here to basically slow the process down. So I guess the way you can think about this is if this was a hydroelectric dam, so this is the dam wall, and this is the water flowing through the dam wall. if there's a problem where, say, the electrons here, the water can't flow appropriately, there leads to a whole bunch of signals in the dam to like slow down the flow or adjust the processes, or if the pressure adjusts at the dam, to adjust the processes to help relieve the pressure, or to help prevent perhaps the water wheel in the dam from generating too much electricity too fast. So the dam will sense, or the proteins will sense that pressure inside the dam, and then they'll start to adjust the function of these like outlets for the water to release, to basically say, oh, we don't want the rotors here to spin too fast, so we're going to shut this piece down, and then maybe we'll slowly let things trickle out when the pressure has been adjusted to some extent. It's not a perfect analogy, but I think it's an easier way to understand it if you don't have complete view of the mitochondria. So essentially, the mitochondria are sensing the electron flow, and they're sensing the amount of, it's getting a sense of electron flow and reactive oxygen species production, which is tied to the electron flow. And then if there's changes in those components, it adjusts these different protein structures, and they will turn on or off their function, or modulate their function in some capacity, so that the ROS levels can be tapered down, up or down, depending on what's going on, and protect the cell from too much ROS. So you can see how there's like, these are fine-tuning mechanisms involved in the cell, and it's not necessarily a hormetic thing. It's more so a signaling component. Okay, so with that in mind, where are the major sites of mitochondrial ROS production? So here we can look at this. We can see the main sites of ROS generation are respiratory complexes 1, 2, and 3. They give the different names here. And so what we can see on this graphic is you have complex 1 here, you have complex 2 here, and you have complex 3 here. And you can see these red arrows with the oxygen creating these reactive oxygen radicals. These are the different sites where all of this can occur. So those are some of the major sites where these reactive oxygen species are occurring. Now, another piece to continue laying the framework here. Coenzyme Q, this enzyme here, is extremely important in this entire process, because when electrons are transferred into the electron transport chain, they all have to largely flow through coenzyme Q to a large extent. So here we have complex 1, and then we have complex 2, complex 3, complex 4, complex 5. Complex 5 is is the ATPase, or ATPase, where you get the production of ATP from the protons moving, following their gradient essentially, and taking the ADP and converting it to ATP. But in the rest of the process here, you have electrons flowing from NADH and FADH2 through complex 1, 2, 3, and 4, and pumping protons across this gradient, and then eventually the electrons are attached in with oxygen to create water down here. So there's a start point for the electrons to flow through. You're essentially generating electrical current. And then what happens is, at this point here, the electrons have a final electron acceptor, and that current is kind of terminated, and you're creating a gradient in the meantime. And so this gradient then powers the production of cellular energy. Now, CoQ10 is pivotal, or coenzyme Q is pivotal in this entire process, because complex 1 and complex 2 both use coenzyme Q to transfer electrons into complex 3. So the electrons have to move through coenzyme Q. There's some other things here that can drop off electrons. Well, it's kind of ancillary to the overall picture, so we won't get too much into those. But essentially, coenzyme Q is key, and we'll talk about why this becomes important as we move along. But think of this as kind of like a wire system where the electricity is being transported through the membrane, or through these different proteins via electrons specifically. Okay, so reverse electron flow. This is where things start to get quite important, and this piece is where the stress component will tie in directly. So I'm going to read a few quotes here, and then we'll talk about, I'll show you the mechanisms of the reverse electron flow and how this relates specifically to reactive oxygen species production, and why this is important in from a metabolic standpoint overall, and how this ties in with stress and different disease processes. So they say here, until recently, RET, or reverse electron flow, was considered as an in vitro artifact with no relevance in vivo. So in vitro is just kind of like in a petri dish, whereas in vivo is things happening inside the body. So they say, however, in the last 10 years, several groups have independently published evidence that does show that reverse electron flow or transport occurs in vivo, and that it has a central role in many physiologic processes, including aging. In 1961, Chance and Hollinger described a previously unknown mechanism by which isolated mitochondria produce NADH from NAD+ after supplying them with succinate, a complex 2 substrate. Later, it was shown that complex 1 reduces NAD+ to NADH, with electrons received from ubiquinol, or coenzyme Q, via reverse electron transfer. Interestingly, reverse electron transfer is associated with the generation of high levels of reactive oxygen species. Now we know that reverse electron transfer occurs when the pool of coenzyme Q becomes over-reduced with electrons from respiratory complex 2, aka succinate dehydrogenase. So essentially, and I guess we'll read here because this will actually tie it all together. Oxidation of glucose produces NADH and FADH2, with 5 NADH molecules being oxidized for each molecule of FADH2 by the respiratory chain. When fatty acids are oxidized, the ratio of NADH to FADH2 changes to 2:1, which results in an over-reduction of coenzyme Q pool and an increase in reverse electron transport ROS, or reactive oxygen species. So what exactly is going on here? So you have, this is complex 2 and this is complex 1. What happens is when carbohydrates are burned, NADH and FADH are produced, except higher amounts of NADH are produced than FADH2. So you have a 5:1 ratio. Now, when fats are burned, NADH and FADH2 are also produced. However, the ratio of NADH to FADH2 is different. So you have a 2:1 ratio. So you have twice as much NADH as FADH2 with fat oxidation, where you have 5 times as much NADH as FADH2 with carb oxidation. Now, what's the problem with this? So FADH2 flows through or drops its electrons off at complex 2, whereas NADH drops its electrons off at complex 1. So when you have a lot of NADH, or the other piece before we get there is complex 1 and complex 2 are sharing coenzyme Q as a carrier to drop the electrons off to complex 3. So when you have a lot of NADH in ratio to FADH2, you have that 5:1 ratio. Coenzyme Q doesn't have quite as much electrons attached to it from complex 2. Again, FADH is dropped off, FADH2 is dropped off here, and NADH is dropped off here. So this leaves a lot of room for complex 1 to drop its electrons off from NADH to coenzyme Q. When you shift that ratio and you're oxidizing large amounts of fatty acids, what winds up happening is complex 2 starts to drop off more electrons overall in ratio to NADH or complex 1, and this coenzyme Q starts to become over-reduced, meaning so electrons have a negative charge, so when they are added to something, they reduce it. And so essentially what happens is the coenzyme Q has a ton of electrons. And so what happens is then this coenzyme Q actually starts to, when it comes over to complex 1, drop electrons off in a reverse flow to complex 1. And what can happen is that can take NAD+, which had already dropped its electrons off at complex 1, and add electrons to the NAD+ to recreate NADH. Now, the other problem is if you have a whole bunch of NADH building up here, and you also have a coenzyme Q that has a bunch of electrons bonded to it, and this complex 1 can't drop off these electrons from the NADH, now you start producing large amounts of ROS, and that's basically what they're seeing here. And so this is, now that ROS is sending a signal to the cell and saying, hey, we have some metabolic issues going on here. There's something going on here metabolically because the amount of ROS that we're producing has changed. We're getting more ROS, and the ROS is now being generated from this particular site of complex 1 instead of this other site of complex 1, or instead of being generated from other portions of the mitochondria. And so this actually signals it sends a message to the cell to adjust multiple factors and multiple components that are going on from a metabolic standpoint, because you have essentially this backlog of electrons coming here and generating a bunch of ROS. So this is why is this important? This is extremely important because one of the main things that triggers this process and this signaling cascade is the oxidation of fatty acids. Because fatty acids change the NADH and FADH2 ratio, it completely adjusts ROS production and signaling at the cell overall. And so what are states where you tend to have large amounts of fatty acid oxidation? Low carbohydrate diets, starvation, and stress. All of these different things are basically triggering the same signaling cascade inside the cells. When you have a lot of stress, you're going to release glucocorticoids, you're going to release glucagon, you're going to release adrenaline, and those are going to mobilize fatty acids and carbohydrates from your glycogen stores, from your fat stores. But as I've discussed in previous videos, and Jay and I have also discussed in Energy Balance podcast, when you are under stress and you upregulate glucocorticoids and adrenaline and these different stress hormones, they also heavily drive fatty acid oxidation. And so they're triggering fatty acid oxidation, and they're liberating fatty acids to be oxidized simultaneously to meet this particular demand. But this changes the mechanics inside the mitochondria and can create these circumstances where you're generating large amounts of ROS, because when you're largely oxidizing these fatty acids, you're changing that NADH to FADH ratio, you're creating a reverse electron flow through coenzyme Q, you're increasing the amount of NADH you have on this side as well, and then you're triggering ROS. So you're changing the NADH to NAD plus ratio as well. So there's multiple implications from this. Now, on the next piece as well is in starvation, the same thing is happening. When you don't have enough food coming in, like total caloric intake overall, what winds up happening is you have to start liberating your body's stores of substrate for fuel, because you're going to need substrate, not for fuel, but to create energy. You're going to need substrate to create that energy. If you don't have energy, you die. So the body makes a decision and says, hey, I'm going to take this fat storage and this tissue that I have. Maybe at some point when the environment gets better, I can actually rebuild those stores. But in the meantime, I need to live. I need to produce energy. So I'm going to upregulate cortisol. I'm going to upregulate glucagon. I'm going to upregulate adrenaline. I'm going to start breaking down protein structure, turning it to carbs. I'm going to start liberating fatty acids. And this happens especially once you clear through all your glycogen stores. And then I'm going to start heavily increasing fatty acid oxidation across the vast majority of the body, except for certain tissues that I've discussed in the past, like, for example, the central nervous system, which will still oxidize carbohydrates. So we're going to shift most things to fatty acid oxidation. The central nervous system, there'll be some ketones there and some carbohydrates available for the central nervous system to function. And then what we'll wind up doing is will change what's going on in the mitochondrial system and the whole entire metabolic apparatus, because when we start oxidizing all these fatty acids, we have the reverse electron transport, and then we have the ROS generation, which sends a signal that we're in these particular states. So in these different states, what you're essentially seeing is that the fatty acid oxidation and the stress pathways are functioning very directly through signaling on the energetic level. So a lot of direct signaling is coming from how much ROS is being produced in the mitochondria, what's coming in from the environment. So you have these multiple levels of fine-tuned regulation going on and adjusting what's going on in the system across the board. So you have the hormonal effects, which are adjusted by your dietary intake, and then you have the effects going on at the actual cellular level inside the mitochondria, and then you also have genetic signaling as well. So there's multiple layers overlapping each other, controlling metabolic rates and whatnot. But I think the reason I'm bringing this up is because I think that as research continues in this area, what you'll start to see is that metabolism is actually a key piece underlying all of this. And some of the ROS signaling in terms of amounts and location and whatnot are going to be sending signals to the mitochondria on how substrate is actually being turned into energy, and basically saying like, hey, do we have a lot of fats? Okay, we're generating more ROS. Okay, maybe we have to adjust the metabolic rate to some extent, or we have to adjust this switches the substrate or adjust insulin signaling. So this is bringing into center focus metabolism as one of the key drivers through these different signaling mechanisms overall. Now, this also is extremely important inside cardiovascular disease and diabetes, and metabolic syndrome and obesity, and pretty much all disease processes, because all disease processes are heavily involved with reactive oxygen species and with substrate oxidation. So like what type of substrates are being oxidized. So for example, in heart disease or in the diabetic heart, you see an actually over-reliance on fatty acid oxidation with an inability to properly oxidize carbohydrate effectively. And I did discuss that in my last video, where I went through the interaction of ROS and polyunsaturated fatty acids in heart disease. So in diabetic hearts or in heart failure, the heart is unable to effectively use carbohydrate, and it relies more and more on fatty acid oxidation, and there also is larger and larger amounts of ROS signaling. And this pathway that I'm describing here, with reverse electron transport flow by changes in FADH2 and NADH ratios, is one of the key mechanisms driving these different processes and in all the signaling cascades that are following that. So this is a central piece inside these disease processes as well, across the board. And then I guess a larger view you can see from this overall, like if we took like a 30,000 foot view, at the end of the day, you can't do anything without energy. So if we put this into modern day life, without electricity, without fire, without gasoline, cars don't drive, lights don't turn on, air conditioning doesn't work, refrigerators don't work, food can't be cooked effectively. So when there's no energy available, the systems completely degrade. And this ties in with Dr. Pete's hypothesis that energy and structure are interdependent at every level, or interdependent at every level. So as energy increases, structures and opportunities are able to complexify. And as things complexify, they require more energy, they can use more energy, perhaps the ability to generate energy increases. And so it would make sense with this perspective, this theory, and these understandings that at the end of the day, energy is central to all of these processes, because without the energy, none of this stuff could function appropriately. So the systems are going to fine tune what their functions based on the amount of energy that they have available, and how do they signal energy availability, particularly in the mitochondria, is you can signal through reactive oxygen species site of production as well as amount of production, kind of like you're looking at the engine, where you're starting to say, hey, the engine is it's changing its oxidation of the fuel, and now we're getting more smoke production. That would be the ROS. And so maybe something's going on with the spark plugs, maybe something's going on with the fuel injection, all of these different components, and that also ties directly in here with the mitochondrial function. So I think this piece is exceptionally key, and I wanted to highlight it because mitochondria, again, are one of the main sources of ROS. You're seeing ROS across all the disease states, but then metabolic changes are also seen across all the disease states. So I think that the metabolism and understanding ROS as a signaling component is extremely important context in looking at these disease processes overall. So tangent aside, I want to jump into the next piece here. So we discussed that reverse electron transport flow can lead to electron leak and an ROS generation at complex 1. So I want to look specifically on how this is happening. So there's two quotes here. They say, one important question that needs to be addressed is where electrons leak to produce ROS within complex 1. So they say 7 iron sulfur centers transfer electrons between the flavin mononucleotide site, so the IF site here, and then where NADH is oxidized to NAD+, and then the Q binding site, so where coenzyme Q binds, that's the IQ site you see here, where electrons are finally transferred to ubiquinone. So the IF, IQ, and the iron sulfur clusters have been proposed to produce superoxides. So superoxides are reactive oxygen species. This can get converted through the antioxidant enzyme systems to hydrogen peroxide. So this is from the other graph. You can see NAD Ideally, it should actually be functioning the other direction, but I snapped this section of the picture from the dysfunction where NAD was being converted to NADH because it's an over-reduced CoQ, dropping electrons off in reverse. But essentially what you're seeing here is you have the IF site, you have the IQ site, and then there's the iron sulfur clusters that run from the IF to IQ site. Think of it kind of like a wire. So you have this place where the electricity is plugged in. So here's an outlet, and then there's a wire, and then the extension, the male piece in the wire that would plug into the wall would plug in the CoQ10 and drop the electricity off here. The problem is when CoQ10 has too much electrons, now you have an electric current moving this way and an electric current moving this way, and then you start getting this ROS production inside the mitochondria because you have basically like almost two competing currents running into each other. The other thing to keep in mind is if this membrane is filled with a bunch of polyunsaturated fatty acids, particularly heavily polyunsaturated or highly unsaturated fatty acids that have a lot of double bonds, well, they're not going to really react well to those that ROS generation, the place where these two currents are meeting and leaking a whole bunch of electrons and whatnot. And that's where they can start to get attacked and damaged. And that's why having the structure maintained appropriately is important, as well as how the energy is flowing through this structure is also important. So I wanted to just give you guys a better view of what's going on. So I took that other graphic above and cut it up a little bit to make this. So you have the IF site. So this is the flavin mononucleotide site. So you see the FAD. This is where NADH drops off electrons to NAD+. And then you have the iron sulfur clusters. And so this is essentially that wire. And then you see the electrons flow across that wire to the IQ site. So this is the site where coenzyme Q picks up the electrons. And so you can get a sense, this whole thing is obviously complex 1. And so this is how the process is working. So electrons are dropped. So this is an outlet. NADH plugs into that outlet, drops the electrons off. They move across this nice wire here. And then at this site here, there's a male piece on the, like kind of the piece that you would plug in. And the coenzyme Q gets plugged in here at the IQ site to take these electrons and transfer the current across the system. So if there's a problem, if you have CoQ10 that has too much, like the current is moving reverse through CoQ10, and the male piece is trying to plug into this outlet that has a current moving in the opposite direction, you start to see these electrons get leaked. They start to leak all over the place because you have two opposing currents. Or if the CoQ10 plugs into the IQ site and the electrons start moving in reverse, but you have NADH here trying to drop off electrons, then you can also get the electrons leaking from this side as well, or somewhere along this iron sulfur cluster. So those are going to be the spots that make the most sense. The other thing is you can also, if you have a bunch of NAD here, the other idea is that coenzyme Q could bind in here, drop electrons off, and then the NAD can actually pick up electrons off of this flavin mononucleotide site. That was one of the things that they were just discussing in some of those quotes. So let me close this one. Now, what's normal forward electron flow with ROS signaling, and then also what's going on with the reverse electron flow. So let's compare them a bit. So we saw what reverse electron flow is. Now I want to just show you guys what normal forward electron flow is. So what they say here is complex 1 can produce ROS when electrons circulate in the forward or reverse direction. When complex 1 linked substrates, so glutamate or pyruvate, in combination with malate, are used to feed the respiratory chain, electrons circulate through complex 1 in the forward direction. In these conditions, blocking the IQ site with rotenone, so that's a mitochondrial poison, causes accumulation of NADH and over-reduction of the IF site, where electrons leak to reduce oxygen to superoxide. This electron leak and subsequent superoxide formation is independent of both the redox state of coenzyme Q and delta P. So let's look at this. So when you drop off, so one of the substrate they mentioned is pyruvate. So pyruvate is generated from carbohydrate or glycolysis, so splitting of glucose or fructose. So it's through carbohydrate metabolism. And so pyruvate's generated, it moves through the Krebs cycle, it produces acetyl-CoA, but it also, it gets turned into citrate, runs through the Krebs cycle, and then it doesn't necessarily produce acetyl-CoA. What it produces is the NADH and FADH2. That's the most important piece to understand here, And so when you get but the thing about the pyruvate and the carbohydrate oxidation is you get a lot more NADH, instead of in ratio to FADH2, what I was just describing. So it's a 5-to-1 versus 2-to-1. And so the NADH, that ratio allows the electrons to move in a forward direction instead of a reverse direction. And so what happens is the NADH drops off the electrons at the IF site. It moves across that iron sulfur wire we talked about, goes to the IQ site, and then coenzyme Q picks up the electrical current or the electrons from the IQ site. It brings them to complex 3. Complex 3 moves these electrons across cytochrome C. And then you have complex 4 here, takes those electrons, adds them to oxygen, and creates the water here. Now, as it moves across this pathway, complex 1 pumps some protons. You see them here. Complex 2 pumps some protons. Complex 3 pumps some protons, and complex 4 pumps some protons. And then so essentially, it's like a electric dam. These things are pumping the protons across. And as the protons are building up in pressure here, they move through this channel here, which is complex 5 or ATPase or ATPase. And then the protons are able to then flow through complex 5. And think of there like a little water wheel in there. And as they flow through, they turn the water wheel. And then the energy that's generated from that can create ATP from ADP. So it will add a high energy phosphate group to the ADP to form the ATP. Now, in forward electron flow, when you add rotenone, this mitochondrial poison or rotenone, however it's pronounced, it blocks the IQ site. So coenzyme Q can't come in and pick up the electrons. And so there's a backlog between the IF and the IQ site. So when NADH drops off these electrons and produces NAD+, the electrons have nowhere to go. So they just start producing, they start being released and creating reactive oxygen species. And so it's blocking from this perspective. The FCCP thing, it's related to adjusting the proton motive force, so the concentration of H+ ions and inside this space here. This is the inner mitochondrial membrane, and this the outer mitochondrial membrane, and this is the space between. So this can kind of dissipate the proton motive force. I don't want to cover that too much because it gets a little bit more complex, but just for the general idea, the electrons move to this IQ site, they're blocked by this toxin, and then they start leaking from the IF site, creating ROS. So this is forward electron flow functioning normally, and then this is forward electron flow blocked by this poison rotenone. Now we're going to talk about why this is important in our next piece. So in reverse electron flow, when complex 2 lake substrates like succinate are used, a portion of the electrons flow through complex 1 in the reverse direction. ROS produced in these conditions depend on the redox state of coenzyme Q. So does coenzyme Q have a bunch of electrons on it? Is it heavily reduced? And then the delta P, which is the proton motive force. So the change in how many protons are here versus here. With both rotenone and FCCP significantly reducing ROS produced via reverse electron flow. It's not clear from where electrons leak during reverse electron flow, although IQ has been proposed as the most probable site of electron leak. So this is interesting. In reverse electron flow, rotenone actually blocks ROS production. So let's see why. So what winds up happening is in reverse electron flow, you're oxidizing a bunch of fatty acids. You get a large amount of production of FADH2 in relation to NADH. And at complex 2, that FADH drops off a bunch of electrons on coenzyme Q. Coenzyme Q is loaded up with electrons. It's like a bus that's completely full. It can't take up any more electrons. And so it comes over to complex 1 to try to get some of these electrons. But the bus is completely full. So all the electrons here, all the people here waiting to ride this bus get angry and they start forming a riot. And that's the ROS. So essentially the NADH drops off the electrons at the IF site. It moves across that iron sulfur wire to the IQ site, but it can't drop the electrons off at coenzyme Q because coenzyme Q is already loaded up. And so you start getting basically a current coming this way and a current coming that way, and boom, you start getting this ROS production. Now, what winds up happening is if you introduce rotenone in these circumstances, Coenzyme Q, and by the way, this creates a crap ton of ROS, like a lot of ROS. And what they're comparing here is ROS amount. So in this other situation where coenzyme Q is heavily reduced and it has a bunch of electrons on board, when rotenone comes here, it blocks coenzyme Q from having this current meet the current coming from the electrons coming off NADH, and it lowers the amount of ROS produced. So in this context, the rotenone is actually protective. And the reason this is very important, I know it's kind of, oh, who cares if no one's going to take rotenone. The reason this is important is because it indicates that it's validating the hypothesis to some extent. Where you're seeing, why is rotenone creating a lowering ROS in this circumstance, where in this other circumstance it actually increases ROS, and it's about how the electrons are flowing. And this is very important to understand overall, because this is helping us to understand in different contexts, electrons are produced, or electrons are leaked, and ROS is produced at different sites and in different amounts. And the different sites and different amounts of the ROS production lets us know what's going on with metabolism, or lets the cell know what's going on with metabolism, so the cell can effectively adjust metabolism to manage to manage it appropriately. So it's part of that signaling cascade, and the rotenone is an example of this for us to see overall, and it further solidifies the idea that the ROS is signaling what exactly is going on inside the mitochondria. It's telling it there's a feedback system where the mitochondria is sensing ROS production at different sites and in different amounts, and then saying, oh, we have ROS coming from this site and this amount, so it means X, Y, and Z things, so we need to adjust in X, Y, and Z way. So it's actually a very intelligent system. It's very interesting overall, at least for me, hopefully for you guys as well. So they go here to say, these results resolve many contradictory findings regarding the effect of rotenone on levels of mitochondrial ROS. As Guaras and colleagues show, rotenone increased ROS when cells are fed with galactose, so carbohydrate, where ROS are produced in the forward direction by complex 1, and reduce ROS when cells are fed with fatty acids that stimulate production of reverse electron transport ROS production. So when the cells are fed with fatty acids and you get the reverse electron flow because of the change in the NADH and FADH2 ratio, then the rotenone is protective of excess ROS generation. But when you feed the cells carbohydrate and the rotenone blocks the ability of the carbohydrates to move electrons forward to coenzyme Q, because again, it's blocking at this IQ site here, then you start to get a bunch of ROS generation. So changing the substrate and then introducing rotenone changes the effects that are seen overall. And I'm pretty sure rotenone is a toxin that causes Parkinson-like symptoms because it directly damages neurons within the central nervous system, which don't oxidize fatty acids. So rotenone's not protective I'm not saying to use rotenone. I'm just saying it's here as an example overall. Okay, so what does this all mean? So site-specific ROS generation as a signaling function. So I'm going to hammer this point home again. So ROS play an essential role in redox signaling. As we previously mentioned, the dominant paradigm is that the amount of ROS determines physiologic effects. However, in addition to the amount of ROS, the site at which ROS are generated is also important. So initially it was just, if we have a lot of ROS, it damages the cell. But a little bit doesn't. And then here it was, now what we're seeing is, ROS location is actually super important. Where's that ROS being generated? And the reason I'm highlighting this piece is because I think it blows the hormetic idea out of the water to some extent, because now what you're seeing is, if ROS is generated in a particular area, in a particular amount, it's telling the cell, hey, this is the type of metabolism that we have going on via interaction with those different protein structures that I discussed, the cysteine groups on the peroxyreducin proteins. And so essentially this paradigm is actually another layer in understanding what's actually going on with ROS. And is it just blanket negative? Is it good in small amounts and then bad in higher amounts? And it's like, no, it's actually the ROS is telling the cell, the mitochondria, hey, this is what's going on with metabolism. So it's not the hormetic idea. It's more so like it's a fine-tuning mechanism. At least that's my take when reading this. The next piece they say here is an interesting study from the laboratory of Stefan Droz demonstrated that how ROS are generated, forward or reverse direction, at complex 1 affects how several proteins are oxidized. This supports the existence of site-specific ROS signaling that activates specific pathways by altering the redox states of specific proteins. This means that stimulating the generation of ROS at specific sites and preventing electron leak from others could have therapeutic effects. Following this, Martin Brand's group has started to design antioxidants that target ROS produced exclusively by complex 1. So basically what they're seeing is that if ROS is generated at different sites, so whether the IF site or the IQ site or whatnot inside complex 1, then different proteins get oxidized, and when those different proteins get oxidized, it sends a different signal to the cell. So in forward electron flow, when you have a lot more NADH, FADH2, there's lower ROS production, and where ROS actually gets produced is not the same as when you have reverse electron flow, where you maybe have more ROS generation, and where that ROS is actually being produced could be it could be a different place, and then that it changes or oxidizes different proteins at that different place, and that sends an entirely different message to the cell. Now, the one thing that I want to say about this is the idea that we're going to find specific antioxidants to target ROS produced exclusively at these different sites is a bit reductionistic. It's like, oh, well, if we put antioxidants in this place and not in that place, then we'll have more ROS there, and then we can control the cell. But I think what makes more sense is, how is the cell metabolizing, and then what does the ROS say about that type of metabolism, and trying to view it through that lens, so that when you're saying, oh, when we are oxidizing carbohydrate and we have this forward electron flow and we don't have an excess fatty acid oxidation, then perhaps we need to just get to a point where we can oxidize a carbohydrate effectively, and then we don't need all the antioxidants in this particular reductive piece or that particular reductive piece, because things are functioning in the mechanism that they're supposed to function. So that would be the way that's the way I would try to go about it. However, if you have somebody who has dysfunction, then it may be necessary to actually target specific areas to help get the system back online to some extent. I don't know. This is newer research, so we're still figuring it. It's still being worked through, and I'm going to be following it and thinking about this continuously as I go along. But I'm thinking that it's more about how electrons are flowing through the electron transport chain and how the energy is being produced, and then how that changes the ROS production and the proteins that get oxidized, and less so trying to reductively manipulate the different areas specifically. It's just get things to flow in the right direction, and you don't even have to worry about it. Which goes with Pete's ideas, or the bioenergetic view overall. So I also created a little graphic for this so you could see. So where the ROS is produced and how much ROS is produced in those different places will lead to specific ROS protein interactions. So specific proteins will be oxidized or by the ROS, and then essentially what winds up happening is that will send a different signaling effect to the different parts of the cell. So this ties in. The next piece is that the reverse electron transport, the specific ROS protein interaction, is actually a tuning mechanism for the cell to adjust what's going on metabolically based on what it's seeing with the changes in these protein structures. So they say reverse electron transfer ROS is particularly well situated to detecting metabolic changes which require metabolic adaptations. Reverse electron transport is sensitive to changes in the redox state of coenzyme Q, which is directly related to electron flow through the respiratory chain, and variations in proton or delta P, which is proton motive force, that relate to the capacity to produce ATP, or so a change in protons across the membrane is delta P. In addition, a considerable amount of ROS is produced from a reverse electron transport. These reverse electron transfer ROS could act as a signal whose intensity could be modulated. So essentially, all they're saying here is that reverse electron transport is likely a signal to the cell, and it signals through these larger amounts of ROS being produced in specific areas. And it's basically what you're seeing is that the reverse electron transport is changing the reductive state of coenzyme Q, which I said, as we discussed, was extremely important because it's where all the electrons have to flow through inside the electron transport chain from complex 1 or complex 2 to make it to 3, 4 to complex 3 and 4. And so the ROS generation and then the subsequent oxidation of those proteins can basically tell the cell like, hey, we have this substrate coming in. We have a lot of fatty acids coming in, so we need to change X, Y, and Z thing. Or we have there's some type of dysfunction going on, so we have to change X, Y, and Z thing. So I think it's likely that the amount of ROS that's being produced in different areas will change how the cell affects like a whole host of gene pathways and different components to adjust metabolism to fit that context. So it's a way for the cell to adjust overall its entire context and say like, hey, the environment is providing us with this substrate or this particular context overall, and so we need to adjust our metabolism or our function in this type of way. So it's a delicate way for the cell to adapt itself to the context which it finds it's in, or the variables that it's being exposed to. So this is a direct mechanism to do that, which is really awesome. So there's a little graphic here where the ROS is produced and how much is produced will adjust what proteins and how much of those proteins are oxidized, and then the cell will use that to fine-tune its metabolism, or its metabolism as well as other signaling mechanisms overall. So now, putting it all together. So we see here that the quote says, mitochondria have evolved several ways to communicate with other cellular components in order to supply appropriate amounts of energy and essential cellular components, e.g. iron sulfur clusters produced within the mitochondria. The entire set of molecules that participate in cellular signaling is not well defined, but a number of transcription factors, respiratory chain components, Krebs cycle intermediates, and of course ROS have been shown to be able to signal for mitochondria. ROS's signaling molecules is particularly complicated since they can cause oxidative damage. It is now widely accepted that ROS participate in signaling at low levels but cause damage at higher concentrations. However, new evidence indicates that the site at which ROS are generated is also important in determining the effects of ROS. In fact, it follows logic that ROS involved in signaling are produced in specific locations at specific times. So again, context is really important. This pathway would rely on redox-sensitive proteins located in proximity to the site of ROS production to relay complex information. From this point of view, reverse electron transport ROS signaling is an ideal system since it detects changes in membrane potential and electron flow, producing significant amounts of ROS exclusively at complex 1. So basically what's going on here is the mitochondria are they send out different signaling components, and they're adjusting their function based on a variety of different factors, including transcription factors, respiratory chain components, Krebs cycle intermediates, so citrate, succinate, malate, acetyl-CoA, all these different things, and of course ROS signaling. And so with the variety of these different things coming in to the picture, the mitochondria are then adjusting function and telling the cell, hey, I need this. Hey, I need more iron and sulfur to build these iron sulfur clusters. Hey, I need more, all these different components. So this delicate interplay where the mitochondria is receiving all of these signals, adjusting its function, and then interacting directly with the cell and saying, I need this, I need that, I need this, I need that, etc. The other thing that they talk about here is that ROS generation is a key piece in this, particularly reverse electron transport, because the reverse electron transport, because of the effects that it has and how it's functioning and the context that it needs in order to actually generate its signaling, it's saying, hey, we have a lot of protons on one side of the membrane. We're not really producing as much ATP and we have a whole bunch of ROS. What's going on? And basically the way that the cells knows that that's actually going on is you get all of this ROS generation at complex 1 that's changing the protein structure in complex 1. And then by the change in protein structure, you have a change in protein function. And with that change in function, there's a whole host of things that can occur afterward. So it's an intelligent process and working through these multiple different steps. So I created this graphic to kind of show the general idea here. So you have ROS location and ROS amount changing what's going on with the ROS interaction with different proteins. That's affecting the metabolic signaling and function. Then you have mitochondrial DNA and different transcription factors that are also coming in and affecting metabolic signaling. The other thing you have is NADH and FADH2 ratios, which can function through the reverse electron transport pathway. That's adjusting metabolic signaling. You have membrane composition and lipid peroxidation products. So if cardiolipin gets super oxidized and creates a bunch of lipid peroxidation products and from the mitochondrial membrane, and then it starts to create a cascade throughout the cell will sense that through damage to structure and subsequent alteration in function, which will trigger a whole bunch of other cascades. And then that can adjust metabolic signaling as well. Then you have ATP, ADP, and AMP levels. So if you have enough ATP, you probably don't need to further harvest high energy phosphate groups off of ADP to create AMP. And so if you start getting large amounts of AMP, you have a signal for change in metabolism at the cell. So that can affect metabolic signaling. And then you also have things like NADH and NAD plus ratios. So if you have an over-reduced cell where you have reductive stress, you have large pools of NADH and dwindling supply of NAD plus, that will also adjust metabolic signaling. So there's multiple factors that are all working concurrently and adjusting off of each other to adjust metabolic signaling. And ways in which they're working through these pathways is when you have a change in the structure of the cell, you have a change in the function. And then when you have a change in the function, there's like a whole bunch of cascades that occur after that. And to wrap it all up nicely, this directly goes with Dr. Pete's hypothesis that energy and structure are interdependent at every level. So when energy is flowing appropriately, these things are able to function appropriately, and then the structure isn't altered in certain ways that create contingency pathways and backup pathways and things along those lines, so it can function appropriately. But when energy is damaged, when energy isn't flowing appropriately, these are very specific mechanisms that we can look at and say, okay, when these proteins interact with a bunch of ROS, the protein structure is changed, and then that protein structure overall adjusts it's the protein's function, and then that changes a cascade of other things inside the cell. And so that's the initial piece for signaling, but there's, I think, longer term implications if you continually have tons of ROS being generated, damaging protein structure, and then function is continually adjusted down the line. So that essentially wraps it up for this video. Perhaps a little bit more in depth on this one specifically. Hopefully it's a little shorter than the previous cardiovascular one. On the next video, I want to talk about like what do some of these mechanisms actually look like inside some of the cell, inside some bodily processes that we can see overall. And then following videos after that, we can look at, I'm going to jump back into some of the cardiovascular components, talking about elements of PUFA, but I wanted to just discuss this brief tangent on reactive oxygen species and go from there. So yeah, hope you guys enjoyed the video, and I'll see you in the next one.